You can take the nucleus of a raging tumor cell and place it in the cytoplasm of a non-tumor cell, and you get regulated growth.
Right.
That rules out any oncogene or tumor suppressor gene as being responsible for the dysregulated cell growth. And those experiments were done in vitro and in vivo, a broad range of these. That's what brought me to the idea that this cannot be a genetic disease by any stretch of the imagination.
He is a professor of biology, biochemistry, genetics at Boston College. He is known everywhere, and I first began to see him popping up years ago in the thick of the ketogenic craze, and he sort of became kind of like a patron saint, but where he really has made a mark is with his research into cancer. If you don't know his book, here's the title, Cancer is a Metabolic Disease. And if you don't know why that is such an earth-shattering title and concept, you'll find out as we go along. Professor Thomas Seyfried is with us today. Dr. Seyfried, thank you for joining us today.
Oh, thank you very much. It's nice to be here.
I was very, very interested in visiting with you. Again, we've spoken before on a radio show, but one of the things that I think about is that an awful lot of people probably don't know why even that title is so controversial for your book. And maybe before we really go into why, can we even go a little bit more granular? A lot of people don't totally understand what we mean by metabolic and metabolism. Could we maybe start with that and then jump off from there?
Yeah, we can do that. But the full title of my book was Cancer as a Metabolic Disease on the Origin, Management, and Prevention of Cancer. And it was by John Wiley Press. Now, I have to point out that there have been a couple of hijacked versions with my name that I didn't write.
I've seen them.
Yeah, you've seen these things, right? They take my stuff from podcasts and they use AI and synthesize it down.
Yep.
And then they say I wrote it, and I didn't. So I only wrote one. It's John Wiley Press is the book from 2012. And it's actually a mitochondrial metabolic disorder. Cancer is a mitochondrial metabolic disorder. And I think it's important for people to start reading more and more about our mitochondria. It is the essence of our life and our existence on the planet. And when that organelle becomes corrupted or inefficient in certain ways, you can get a whole range of chronic diseases as well as cancer. So it's important to know about that organelle, how it functions, and how it can be disrupted in its function, leading to dysregulated cell growth, which ultimately is cancer. So that's the essence of what we need to know. And that brought us into a very deep dive on mitochondria, metabolism, biochemistry, and all of the different things you need to know about the origin of that organelle and how it participates in our overall general health.
One of the things that is a telltale sign that they have not gotten your book, even though the fake books still have your name on it, is the thickness of the books. Your book is dense, it is well-researched, and it is an easy read, but it requires thought, particularly depending on if it's a rank lay person reading it or not. Would you explain why this is such a controversy and what the predominant theory of cancer has always been that you are challenging with this book?
Well, the book was in 2012, which laid out a pretty solid argument against the prevailing view that cancer is a genetic disease. But since that time, we have published many other peer-reviewed papers in a number of journals solidifying the view that cancer is predominantly a mitochondrial metabolic disease. The controversy involves the continual belief that cancer is a genetic disease, and that's supported by the National Cancer Institute's website, which says that cancer is a genetic disease. It says it right on there. Mm-hmm. It says cancer is a genetic disease. made up of maybe hundreds of different diseases with many, many different kinds of mutations. All of that is nonsense. It's not, data don't support that concept. And I've written to the NIH, they're like bots. I don't think they think. They just do stuff. And it's consistent with what everybody else thinks it should be. So the continual persistence of the view by the federal government and the academic industry and the pharmaceutical industry is that cancer is a genetic disease driven by all kinds of mutations and tumor suppressor genes and oncogenes, and you're constantly chasing these things around for diagnostic and therapeutic purposes, and we're not getting anywhere.
No.
We got 1,700 people a day dying, 70 an hour, and we've been doing the same stuff for the last 50 years, and nothing's changed, and it's getting worse.
Nothing's changed.
Yeah, that's the sad thing.
Well, I asked you before we started recording the show if this has been your prevailing view. I said that you were a professor, among other things, genetics. And if you think of yourself as an expatriate of the genetic model, and you have always, it sounds like, questioned that model for cancer, yeah?
No, I didn't. I just bought onto it like everybody, all the other non-thinkers. So because that's what it said in the textbooks. And when you're teaching genetics out of the textbooks, and you say, well, okay, it's a genetic disease, tumor suppressor genes. And then in biochemistry, you got the same thing. And cell biology, you got the same thing. But I wasn't focusing on that in those days. I was working in the field of epilepsy. So it really wasn't genetics of epilepsy and glycolipid biochemistry. So it really wasn't central to what I was doing anyway. And I just went along with the crowd. If everybody says it must be right.
Hard to push against it.
Yeah. And then when you start pushing against it, you get weird looks by people. You may have lost your bearings.
Yeah. Well, what began your shift into a metabolic model?
Well, it sprang from our work with ketogenic metabolic therapy for managing epilepsy, which is a standard practice for managing seizures in children. The question is, what is the mechanism by which shifting from a regular high carb diet to a low carb high diet that elevates ketones? How does that stop epileptic seizures? I mean, this is still a very active research focus.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
But we also were studying angiogenesis in brain cancer, which is abnormal blood vessels. And we started to see similarities. When you calorie restrict animals with epilepsy, the seizures go down. And when we calorie restricted mice with brain tumors, the angiogenesis went way down and the tumor shrunk. So I said, well, there seems to be something here. And then we ran into the work of Otto Warburg. And he said that cancer was a metabolic disease and that all cancers have this dependency on glucose because respiration was inefficient. So we started looking at blood sugar and ketones in the animals that were restricted. And sure enough, blood sugar went down and ketones went up and the tumor shrunk. And then Warburg said this a long time ago. That initiated probably around the year 2000, 2001. Brought my attention to that. And then when we started doing research in the lab, we were corroborating what Otto Warburg had said. Then I started to do a real deep dive on Otto Warburg. Who is this guy? Of course, he turns out to be one of the giants in the field of biochemistry.
Giants.
And, a very arrogant German who actually he thought he had all the answers to the cancer problem. And in the end, they threw his ass under the bus.
Uh-huh. Yeah, well, and part of the work that you've done that I think is every bit as profound as laying out the metabolic model. There's work that the way I think of it, Professor Seyfried, is you've stood on the work of Otto Warburg. You've seen what he got right, but you also saw something that he got wrong. I believe you and a colleague from Budapest have published on this. Talk about the additional thing that you guys have found.
Yeah, well, he got something wrong, but he also did not know about something else. So there's two issues here. The first issue is that he assumed that every mole of oxygen consumed by either a normal cell or a tumor cell was comparable to 7 moles of ATP produced, which is the currency of energy in our cells. And he got that calculation from Hans Krebs of the Krebs cycle. So, and Krebs was one of Warburg's students. So because Warburg trained Krebs cannot be wrong. So, so we started to look deeply into, and of course, everybody was saying that Warburg cannot be right because there are some tumor cells that consume oxygen just as avidly as normal cells, and normal cells consume like tumor cells. So he says they were using oxygen consumption as an accurate surrogate for ATP production through oxidative phosphorylation. We and others have done some really comprehensive experiments to show that in cancer cells, oxygen consumed is not quantitatively linked to ATP produced. It is in normal cells for sure, but not in tumor cells.
Not in tumor cells.
Yeah, so tumor cells consume oxygen, but they use the oxygen for the production of reactive oxygen species, which are carcinogenic and mutagenic.
Yes.
So you cannot quantify and make an assumption that the oxygen consumed in a tumor cell is equivalent in energy production to oxygen consumed in a normal cell. And Warburg made that mistake, and everybody make that mistake, and they still make that mistake. They're still using all of these devices like the Seahorse. They're spending $75,000 on the Seahorse instrument, which is all calculations and not direct measurements anyway. So
I didn't realize that. Really?
Yeah, wow. You're not doing direct calc. It's all based on adding these different chemicals, and it's over a short term, like 30 minutes of experimentation, maybe an hour at the most. You got to look at these tumor cells over hours. We use a Resivir oxygen consumption device that we have here in the lab, and we can monitor all over days oxygen consumption on these tumor cells. So we were finding that in normal cells, if you add cyanide, which blocks cytochrome C, oxygen consumption goes way down. So we were giving cyanide, potassium cyanide to the cancer cells, and oxygen was going way down, but ATP wasn't.
Interesting. Right. So there's no respiration going on.
Cancer cells can live in cyanide. They can live without oxygen. So then what we did is we put them in glutamine alone with no glucose and no oxygen. And then we were measuring ATP production. And where was this ATP production coming from? It's coming from mitochondrial substrate level phosphorylation, which is a very interesting mechanism inside the mitochondria itself that produces ATP independent of oxygen. It's unbelievable. So
interesting.
Yeah. So this is where they're using a fermentation mechanism in the very organelle that's supposed to get energy from respiration.
Wow.
So this is an ancient, we have a big paper coming out on this. And this, let me give you an example. When people have heart attacks and they stop breathing, the bloodstream fills up with lactic acid and succinic acid. This is because you're not breathing. And it's been known for a long time that lactic acid is coming from the fermentation. It's called lactic acid fermentation using glucose as the fuel. And Warburg knew this. And that was one of the interesting things about cancer. They continue to ferment even in the presence of 100% oxygen. But he never knew anything about the succinic acid. So we started looking into the guys in the heart attack field knew it all. They knew it. And they were saying that this succinic acid is coming from a amino acid fermentation, and that amino acid seems to be glutamine. So we added labeled glutamine in cells that were no oxygen and no glucose, and we were finding labeled succinic acid. That's the waste product of mitochondrial substrate level phosphorylation. All major cancers are crapping out lactic acid and succinic acid, the two fuels driving the fermentation metabolism. They've been knowing, and they do it in 100% oxygen. So, in other words, they're not respiring if they're fermenting even in the presence of oxygen. So, Warburg knew the lactic acid story. He did not know about the succinic acid story, nor did he know about amino acid fermentation.
And that's the key. And this is something that's really important because there are a lot of lay people, a lot of cancer patients who, once they begin reading, they will see the glucose link, but many of them have not heard about the glutamine link. And this is what is so important because I think that there's implications with regard to therapy, even diet, but the glutamine link is fascinating, and I think that it has not filtered its way into the general public yet the way glucose has.
Yeah, well, in the scientific literature, you'll see many, many papers from top journals calling it cancers are glutamine addicted.
Addicted? Yeah,
glutamine, they'll call it glutamine addicted. They know that tumor cells need a lot of glutamine. But here's the situation. There's what we call anaplerosis. This is a term that means the replenishment of carbons to the TCA cycle, the Krebs cycle. And many investigators were under the assumption that the glutamine entering the cancer cell was respired, not fermented, because the cancer cell is taking in oxygen, it's producing ATP in the mitochondria, and glutamine is being used. And they said, oh, it's coming into the mitochondria and it's being fully oxidized. But if that's the case, they should not be ever producing succinic acid as a waste product. So the glutamine is metabolized to glutamate, and then through a transamination reaction, it's metabolized to the metabolite called alpha-ketoglutarate, which is part of the Krebs cycle. Alpha-ketoglutarate is then metabolized to succinyl-CoA, and that succinyl-CoA to succinate produces ATP through a substrate-level phosphorylation reaction. It's unbelievable. Now, in normal cells, it's doing that, but it's a tiny amount of ATP produced in the cycle. In the cancer cell, it's a buttload of ATP.
No kidding.
Produced. Yeah.
Wow.
So the succinic acid should stay in the cycle if it's going to be fully oxidized. But because it has an incapacity of oxidative phosphorylation, it blows the succinic acid out as a waste product of amino acid fermentation, just like it blows lactic acid out as a waste product of cytosolic substrate level phosphorylation. So the cancer cell is getting energy from two substrate level phosphorylation reactions, independent of oxidative phosphorylation, which is inefficient.
Right, inefficient. And to me, what that does is change the game that you and others have already helped to change even further. It moves it further downstream into the metabolic model. It becomes irrefutable, and yet the conventional docs, the conventional researchers that are relying on the textbooks, it's just so hard for them to turn around, it seems like, yeah?
Well, they're locked into molecular biology and gene chasing. And when you're chasing genes, you're not thinking of the biochemical reactions responsible for energy, because without energy, nothing can survive. ATP is the gold standard. So all of the stuff that most people are studying are downstream effects of all kinds of things. All of the hallmarks of cancer are all downstream effects of the shift from oxidative phosphorylation to substrate level phosphorylation. And, the HIF-1 alpha, which is a transcription factor that becomes stabilized and drives the glycolytic pathway producing lactic acid. That Greg Semenza received the Nobel Prize for his work on HIF-1 alpha and how in hypoxia it becomes stabilized to increase cytosolic substrate level phosphorylation as a means to get energy. Well, here's a very interesting phenomenon. When you have hypoxia, you also produce succinic acid through the ferment. Like I said, the heart attack patients are dumping out succinic acid. Succinic acid also stabilizes HIF-1 alpha, thereby driving the fermentation in the cytosol. So it's our view now that the key to cancer is mitochondrial substrate level phosphorylation, producing succinic acid, which then stabilizes HIF. So HIF again is another downstream effect of mitochondrial substrate level phosphorylation.
Another downstream effect. You've mentioned mitochondria. Let's talk about that for a minute. You know, the evidence that you've compiled and published and written about is really talking about what happens when mitochondria fail or break down. And it shifts from an efficient energy production to a backup primitive function vis-a-vis fermentation. One question that's very basic is, what is causing those mitochondria to fail in the first place?
Well, that was Albert St. George's oncogenic paradox, which we solved. We solved the paradox because people were always asking, what is the in fact, if you look at Sid Mukherjee's book, The Emperor of All Maladies, which was a New York Times bestseller, he asks in there many, many times, what is the common pathophysiological mechanism that could link all of these disparate cancer-causing agents to a dysregulated cell growth? And Albert St. George asked the same thing, and he called it an oncogenic paradox. So what we did is I went through all of the things that we know have been linked to cancer, like carcinogens. When you say, what is a carcinogen? A carcinogen is any chemical that has a potential to cause dysregulated cell growth, which is cancer. But how does a carcinogen cause cancer? Well, according to the somatic mutation theory, it causes DNA mutations. And that's how they think you get dysregulated cell growth. But you also find that many of these carcinogens enter the mitochondria and they biofluoresce and they damage oxidative phosphorylation. So you have this conundrum then. Well, which is the chicken and the egg? Does carcinogens cause cancer because they cause mutations in the DNA? Or does it cause cancer by disrupting oxidative phosphorylation, leading to a compensatory substrate level phosphorylation? Before I get into the actual nitty-gritty of the mechanism that happens, you have to then look at radiation, intermittent hypoxia, inflammation, rare inherited mutations. You have to look at microplastics, forever chemicals. We went through every kind of a risk factor. We found that all germline cancer risk factors all damage oxidative phosphorylation. So the genes are all BRCA1, Li-Fraumeni, Lynch syndrome. We went through, we have a big paper coming out on this showing that all these so-called genetic risk factors all damage oxidative phosphorylation, leading to a compensatory substrate level phosphorylation. But the question is, okay, carcinogens do cause mutations in DNA. How do you know whether those mutations are responsible for dysregulated cell growth? And the answer is, in general, only if mitochondria are damaged do you get dysregulated cell growth. The nuclear mitochondrial transfer experiments excluded genetic mutations as the origin of dysregulated cell growth, because you can take the nucleus of a raging tumor cell and place it in the cytoplasm of a non-tumor cell, and you get regulated growth.
Right.
That rules out any oncogene or tumor suppressor gene as being responsible for the dysregulated cell growth. And those experiments were done in vitro and in vivo, a broad range of these. That's what brought me to the idea that this cannot be a genetic disease by any stretch of the imagination. But here's the key, and this is the golden. All of these downstream effects, like the hallmarks of cancer, which is, angiogenesis, dysregulated regulated growth, and all this. When mitochondria become corrupted and they can't generate energy efficiency through oxidative phosphorylation, as we said, you get a compensatory substrate level phosphorylation, which is replacing the availability of energy from fermentation. Fermentation is replacing oxidative phosphorylation. But that still doesn't tell us. It just tells us where the energy is coming from to drive the dysregulated growth. But where do you get the dysregulated growth from in the first place? Okay. So here's the situation. In the inner membrane of the mitochondria, there's an enriched lipid called cardiolipin. Cardiolipin is enriched in the inner membrane of the mitochondria. It intimately interacts with the proteins of the electron transport chain. It interacts with what we call calcium calcium transporters in the inner membrane of the mitochondria. Okay, mitochondrial calcium transporters are controlled by cardiolipin. Electron transport chain proteins are controlled by cardiolipin. So what we have never found, there's no tumor ever found to have normal content or composition of cardiolipin. So cardiolipin controls calcium signaling. Mitochondria control calcium signaling throughout the cell. The cell cycle itself has checkpoints to control cell division. And those checkpoints are under the control of mitochondria-generated calcium signaling. So when the cardiolipin lipid is abnormal, calcium signaling gets haywire. The cells no longer respect contact inhibition, and it's like the cell cycle no longer has any checkpoints. It falls back on the ancient pathways of fermentation, which were unbridled proliferation. Exactly. Well, you put all this stuff together.
Goodness.
You know,
it becomes a unifying theory.
Absolutely. We now know exactly we know exactly what's causing dysregulated cell growth, why there's no contact inhibition, why they're blowing out fermentation metabolism. And better yet, we know now how to manage the disease very simply.
That is ultimately what it comes down to, is what the implications are both for prevention and for treatment. Again, a lot of people who have been just casually ketogenic for whatever reason have keyed in on a lot of what you're saying. I am wondering first if you have any sense of what the implications are for preventing cancer in the first place, lifestyle changes, dietary changes, etc.
Yeah, well, that's why we developed the glucose ketone index calculator. It was first produced to manage brain cancer, but now we know that all chronic diseases and cancer stem from damage to mitochondrial dysfunction. So if you can keep your mitochondria healthy, the probability of getting cancer is very low. The risk is very, very low.
Incredible.
And also other chronic diseases as well, type 2 diabetes, cardiovascular disease, dementia. The issue about some of these other conditions, which you have mitochondrial dysfunction without corresponding dysregulated cell growth. Usually that happens, the cells die because there are certain cells that can't switch to fermentation because of the types of cells they are, like neurons in the brain and cardiac myocytes. They never become cancer, rarely if ever, because they can't transition to fermentation, so they up and die.
Interesting.
So you know why certain cells can become cancer and why other cells cannot become cancer. So in using a GKI, you always keep low glucose and elevated ketones, putting our cells metabolically back in the Paleolithic state of our existence.
There we go. There we go. All right. I hope that you enjoyed the part 1 to our conversation with Dr. Thomas Seyfried. I'll tell you what, part 2 becomes very, very practical. Dr. Seyfried gets really, really practical. We talk about diet. We talk about exercise. We talk about supplements. We talk about fenbendazole, mebendazole, and these anti-parasitic drugs that so many people are taking and why they seem to be having an effect. This is worth sending to your friends who have an active diagnosis. Again, we are not doctors. We are not giving medical advice. This is just the sharing of information. We're so glad that you watched it. Make sure and send it to your friends. And thank you so much for watching Ultra Life Today.